wondering where Just Biotherapeutics is, if you just look up, we have the whole second floor. So that's why I put just upstairs. Didn't need to tell you we're in Seattle. I wanted to give you a little bit of background so you'll understand the people at Just and how all this started. We actually incorporated back in 2014. It was incorporated. Jim Thomas and Victor Fung and Dean Pettit started the company, and it was a big thanks to them because from there in December of that year, about 10 or 12 of us got around a conference table that was rented, a room down in Pioneer Square, and we literally sat down and said, what are we going to do? And what we wanted to do was really develop innovative technologies so that you could get drugs out to people much more. Because we've worked in the fields. I worked in the biotech field, as she said, for the last 25 years. And many people at Just had worked there, for even longer. And the thing about the biotech is that the drugs are actually very expensive. There we go. And so, the cost, even when you look at the top three biologics that are out there, Humira, Rituxan, Enbrel, you say, well, they're $38,000, $37,000, $20,000 a year, and if we all have insurance, that's great. It pays for quite a bit of them. But for the vast majority of people, even in the U.S., maybe 43% of this is paid for. That can still leave you with a significant cost. And if you're in the population that's making maybe $20,000 to $50,000 a year, you can calculate pretty fast if you have these diseases that need these, that it is a very significant cost that you would have to endure in order to be treated. And with the new precision medicine coming on, it's great medicine, but it's very expensive. And a lot of the diseases now, especially in the infectious disease area, you need combination therapy. So again, that's making to be very expensive. And when we think about these of how do we get to emerging markets? We had our a great talk today, about the Gates MRI and really thinking about emerging markets and low income. And that is probably one of the most difficult areas. We work with the Gates Foundation, and they very quickly told us, you need to develop, we're working on broadly neutralizing antibodies with them. They're these anti-HIV antibodies, and you need to develop those for $20 or less per year. And when you really think about those, most antibodies are costing $200 to $1,000 per year to produce per gram. And that's what I meant before, is $20 a gram to make those. So that's a significant technology innovation that's really needs to be developed in order to get into these markets. So we really started thinking about this, and this is, initially we were thinking about the biophysical and chemical features of the antibodies, and most of my talk is going to really focus on the molecules themselves, but there's a lot more to it. There's really the talking about starting with CHO cell features, because you're taking antibodies, you're taking the sequences, and you're making stable CHO cell lines, and those are used in bioreactors, and then from there you're producing the downstream and purifying the drugs. And that's what we've focused on, for a lot of the people just in our careers. And as we do that and we think about that in the CHO cell, there's a lot of different areas that can really affect that, all the way from how you grow them in the bioreactor, to the media that's involved, to how many times you're adding media to that bioreactor every day. All of these things can get towards the cost of the productivity and the we think about grams per liter per day of material being produced. But then we go into our looking at our downstream processes, and I'll talk a little bit about that later. But everything that we can do to improve the downstream, to improve yields, that you're always trying to push it. How much more can we get out of this plant for the putting in the same cost? The impurity profiles, there's been many times where you see, impurities coming through with different drugs, and they cause huge problems, so we have to look at those. And then finally, when you get to the end, you have your drug product, and you're really trying to think about how do we get that out to the patients. And when you think about the low-income countries that we're working with, you think about many of the countries in Africa and focusing in sub-Saharan Africa, you really want to get out to the outlying areas where you don't have the refrigeration. So we really have to think about how do we improve the molecules to get the stability at room temperature so you can really get out to the patients that need it. And then that always translates back to being able to produce better drugs for here in the US and the developed world. So it really kind of fits together. And then the area that we're really starting to think about now, when you think about lowering the cost, well, you can lower the cost through how much you produce per gram, but you can also lower the cost with the biology. So the areas we're starting to think about is really how do we change the in vivo clearance? Because if you can, you inject a drug and then it's around, say, 2, 3, 4, 5 times as long, you're injecting that much less drug. That lowers the cost right away. And then we're also thinking about many of the drugs now are injected as sub-Q, so you're going into the sub-Q space, and that presents its own unique problems with bioavailability. Many are 50 to 80% bioavailable, so we're thinking about how do we increase that as well. And all of this, in the end, comes down to the amount that it's costing the patient per year, because that's really what's going to drive, can that patient get the drugs? We're trying to put all this together. So the way we did it, and the way we're doing it now, is to really, we thought, well, how do we change the whole paradigm of how drugs are produced? And we went from a large pharma background where you come in, you get your molecule, your process, and everything's siloed. It's a very siloed system, where once the molecule comes through from discovery, then it goes to the people producing the stable CHO cell lines, and it goes to the process group that's going to develop a process for that. I was in formulation development, so it would eventually come to formulation, the analytics. And many of them were, you'd be in different buildings, you'd be trying to work together, and it was actually pretty hard. You'd lose huge amounts of data, and believe me, when we would buy companies, we'd have to actually reproduce exact same studies that were done because the data was just gone. Now with, hopefully having computer systems, it's better, but that still happens. And then this gets punted over to the plant, and then the plant goes and develops its own processes because they need to scale up from 500 liters up to 20,000 liters. And so this just goes on and makes a very slow and costly drug development process. And what we've decided to do, and seems to be working very well, is really to do an integrated design where we have the molecule, the process, the plant, we all sit together, we work together. What we develop as the molecule goes to the process group. we pay attention to what's happening in the plant, and then and this process moves. And what this does is tries to make really predictive models. So for the machine learning and the modeling, that's how we're trying to fit together. And this really goes towards, you'll see 6 areas, but the first 3 is the quality. You can't lower the quality, so and we're always trying to increase that. We're trying to increase the cost, and then it was talked about today, how fast can you get to the clinics? Because that will lower the cost. So we're paying attention to everything. And then the other parts are, once you have your drug, do you have the capacity? And you really want to be very flexible on your facility, because a lot of times drugs start out, you don't need to produce that much. You don't want to have a large facility, so you want to have a facility that you can build up, and we've designed those and actually deployed them, one in China. And you want to be able to go around the world, and then, as was talked about, you'd like to get the drugs produced in the countries that need them so that you're not shipping them, and you're also training the people that are in those countries, so it just all works together very well. So our way to do that is to really, if you think about it, what's the cheapest place to start with? Well, we got the internet, and we've got just hundreds of millions of sequences of antibodies that are available, and those are dirt cheap. It's like you got a computer and you can get them off the internet, you're all set. And so we go in and we've pulled those down, and through homology modeling, we produce the structures. But then you say, well, that's great. What are you going to do with that? Well, what we're trying to do then with that is to then, we've developed a whole series of biophysical assays that I'll go back and explain or I'll be explaining in a minute. But we're actually trying to use those biophysical assays to understand what's going to happen in the process. So as you go through your process and you're developing your cell lines, as you're developing your downstream purification processes, as you're wanting to concentrate them up to high concentrations, as you want to store them, if we can actually predict that based on our characterization assays, and we can do that very quickly, you can pick the best molecules up front so you don't have to do as much process development. And by the time it gets into the plant, you're running along very well. And we'd actually like to skip all this if we could, and you could say, could you give me a sequence? I could then in silico optimize that sequence, jump right into a process and get over to my GMP production and cut out a huge amount of cost and come in with a molecule that's going to work. And that's really where we're trying to do that. So how are we going to do that? Well, we're trying to really do that, and this is where the next whole portion of the talk is going to focus on, is the molecular design. Because the molecule itself is really going to direct a lot of the stability that the molecule has. The sequence is going to, and how it folds is going to direct how it's going to be in the downstream process. We're trying to understand how this fits in to the bioreactor and the production that we get, and all of this needs to fit together. And we've started to have some success with actually trying to have predictive models. So as I said, if you have a sequence, and you can do something like we look at the unfolding temperature at high temp, and that unfolding temperature, as you increase that, generally gives you a more conformationally stable molecule. And for some molecules, we can come in and we can actually predict what that's going to be. So antibodies can be particularly difficult, and at the same time, they can also work very well, as you know, when they're produced, and you go through B-cell maturation, you get somatic hypermutation, which puts in all these mutations into the molecule, and we really focus in on the FV region, and specifically we're looking at the framework regions around the CDRs, and we found that we can predict the sites that have the potential to significantly decrease the conformational stability of the antibodies. We can also go in very quickly and say these are deamidation isomerization sites. You get chemical modification, you get alternate glycosylation, which can be both good and bad sometimes. You get cysteines in there that you don't want. So all of these things can be looked at. We can come in, we can look at the molecules and say, well, which ones are going to be potentially destabilizing? And if you can identify the residues that when you modify them, you've increased the conformational stability, many of the other problems are going to go away. So how do we do this? And this is what we've been doing. We're actually in the process of changing this because now we're gaining a lot of data, as I talked about. But we've come in and we have a process that was developed. It's an in silico tool called Apicus, and it allows us to go in and identify. And in the model here, we actually did identify about 20 different sites. And when you do that, you'd actually like to make all of the changes one time, because we found that the combinational modifications are really what get you where you want to go. And when you do those, you make those, you'd probably be making up into the hundreds of thousands or millions of molecules. And at the moment, we don't have the ability to do that, although we're trying to develop that. So what we did in this case is we came in, we looked at each variant by itself, where we said, well, this residue should be modified to this other residue. And we did that for each one, and at the same, so we had, about 20 different variants, and then at the same time, you always have to maintain potency, and we found these are, looking at BNAbs, is that those are designed to bind to the GP120 of the HIV, and we were told, well, you probably can't change those. You're going to modify the binding. You're going to lose neutralization potency. Well, the fact is we could, and we did, and we moved on from there. From there, when you say the optional second round 2 variants, that's where we actually did come in. We picked 5, made combinational variants, so you'll see where that's going to go in a minute. And then from there, you go through, you do your high throughput biophysics, and we can figure out where we want to be. So this is where we start tying together, what are the parts of the process and how does that tie into the high throughput biophysics analysis that we're going to do? And we're going to collect all this data we've now collected it for thousands of pieces of data here. So we're starting to get to the point of, we have enough for prediction, but not quite. So we look at the bioreactor and the harvest, and what you really want to do is maximize your volumetric productivity. We're not quite there yet, but we're trying to understand that. During your harvest, and this is a very big area, is aggregation of the antibodies, even in the bioreactor, and so we have assays that actually look at aggregation. And then when you're purifying antibodies, you have to undergo a low pH viral inactivation because you're making them in CHO cells, and there could be human viruses that are contained. So you want to make sure those are killed. And actually, there's the chromatograms over here on the right is just an example of this, where we can come in, incubate it at a low pH 3.3, and then neutralize very quickly, and that's where the aggregation happens. And you can see the one in the blue aggregated, that's the parental, and when we do these combinations, we get no change. That's what we're after, so we can do this. And then when we think about the chromatography steps, the viral filtration, and especially when I talk about the formulation, I'll show you later, there we're really paying attention to a number of areas. One is conformational stability. That's huge for the proteins. And then the other is colloidal stability, and I'll go through some of these assays and show you how they work. But this is an example of what we were doing early on, where we had the antibody. This is one of the BNAbs, and it had about 20 different sites that had to be changed. And so we went through, and we looked at each one at a time. And up here in the right-hand corner, I'm going to see if I can do this right. Yep. So up here in the right-hand corner, this is called differential scanning fluorimetry, and this is a very high throughput assay. So everything we do has to be high throughput. I was originally told that well, if you're going to do high throughput, you need to be able to run a few thousand of these in a week. And so if you were ever doing differential scanning calorimetry, you'd go, no way. But with differential scanning fluorimetry, where we're just putting in a dye, you heat it up as it unfolds, the dye then fluoresces, and you can see where your proteins is unfolding. And what we have here is in the blue, that's the MS-194. That's our parent molecule. And normally you have the CH2, and then you have the FAB, and the FAB will unfold at a higher temperature, say 80 degrees. And you can see that occurring with the green one where it says TM2. But in this molecule, we had the CH2 and the FAB unfolding together. That meant that the FAB that we were trying to stabilize was much less stable. So what we did was go in and we looked at this whole series of variants that we had. And we then very simply said which one of the molecules that we produced. We produced we produced up to about 30 molecules for all the variants that we were looking at, and we said which ones have a TM2 because that tells us it's more stable. Well, you could go through and you could look at those and you can say, well, these 5 here all have TM2, and this one out here, which is actually a lower TM2, so maybe it's not as stable. We thought, well, that would be great. Now, what we were losing, you can see this green here versus the red. The red one has a TM2, but it's very small. And so what we realized, we were actually losing a lot of data. And people just go through, and it's really all they look at is TM2. So we developed some algorithms that take advantage of all this data. And that's the one right here. And I'm going to have you pay attention to that later because this is position 108 in the heavy chain. And when we do that, and we did all these combinations, what it turned out was paying attention to that position 108, all of this whole region up here where we're looking at the DSF weighted, we call it the weighted shoulder score to take advantage of all the data down there. And we compare it down here on the bottom, for those of you who can see, that's the chemical unfolding. So we look at, if you've ever done chemical unfolding with guanidine, you know it's a very slow process that can take you a few days. We've developed this so we can potentially, when we have it fully automated, we're going to be able to do a few thousand of these in a week. So this ends up being very important because what we find out is that the chemical unfolding and the DSF together gives you a very good indication that you've conformationally stabilized the protein. And then what goes along with that, here's the pH 3.3, and so you're looking for everything that's dark green. These essentially have no aggregation. Down in the lower left-hand corner, and that includes the parental, those ones were aggregating where you would aggregate about 40% of the protein where it was going through the low pH viral inactivation, which means you're going to lose all your protein when you go through your process. So that's not too good. But as I said, sorry about that, the T108, it turned out if you make this mutation at heavy chain T108, and this is the exact same spot for every antibody because we do this based on the AHO numbering system, which is structurally based, so that's why you can compare across antibodies. And you can say at that position in the framework region, if you change that to an arginine, this worked very well and we stabilized the protein. And the question was, what did we do? Because we can learn that, then we can include that with our machine learning. And what it turns out is that when you look at that, with the threonine 108, there's just 3 hydrogen bonds with this valine across the way here, and so when you heat it up or you bring it into low pH acid, it was very unstable. This is the CDR3, and when you put in this arginine, now all of a sudden we added 4 ionic bonds, we added 4 hydrogen bonds, and we really stabilized the protein. So we can actually not only go in and predict which residues are going to be stable, potentially stabilizing, but at the same time now we're also learning what we're doing so we can go back and apply that. And so this is where the process keeps going around. And that one we actually took through downstream processing, and it just looks great. Now this is another antibody that I wanted to show you, and this one we actually put in 6 mutations. And I want to, and in this one we had 6 mutations in here, and then in another one, Optimize 2, we actually stuck in, normally you see glycosylation in the FAB region of the protein, you don't want that. That causes problems during downstream processing because it's not consistent. So we said, well, let's get rid of that. And so that's why you'll see the Optimize 1 and 2, and I'm going to talk about what the glycosylation did in a minute. But if you come over to the left-hand side, you'll see we're talking about the estimated elimination half-life. Now, we put in the LS mutation, which many people know that will automatically give you an extended half-life in the bloodstream. And when I was talking about that can significantly reduce the cost. Well, that's great. For this particular antibody, looking at a mouse model, it turns out that we only got a very minor increase in the half-life between if we had the LS and not LS. But once we conformationally stabilized the molecule, and that's these 2 box plots up here on the right, we got a significant increase in the half-life. Now this is mouse studies and it's getting ready to go into human studies. But in this particular mouse model, the TG276 model, it's translated very well to what you see in the humans. So we expect that to work well. Now, the other part that I talked about was the formulation, and this gets us into thinking about, can I formulate and put these drugs into areas like sub-Saharan Africa, getting out to the outlying areas where you're not going to have refrigeration. For this particular antibody, the parents, you see this forms a oligomer formation, and as if you get oligomers formed, this may cause immune reactions, in which case it could just take your antibody and your drug out of the bloodstream and basically makes it no good, so you don't want that. But by doing these modifications, you can see over a 12-week period, we barely got about a 0.4% increase. And if you said, well, and this is at 40 degrees. So hopefully, we would say, you're going to limit it to up to 30 degrees, and you can monitor that. But that actually gets us to the point that we can think about getting these outside of the cold chain, and getting them out of the cold chain then gets you to the people that you're trying to get to. So that's kind of a summary of what we're doing and why it's important to do the molecules. Now, the other part that I've talked about is that we really need to pay attention to the process. You see these big manufacturing plants. They are very complex, and we realized early on that we really need to understand all the parts of that process, and because you want to say, where should you focus your efforts? Do you focus it on the molecule? Do you focus it on the equipment? What do you want to improve? And so we have a group that is all they focus on is modeling and everything. And you can see over on the right-hand side, these are all the different areas, the capital materials, consumables. You can break all this down. And then you can break it down into the process. So you've got the bioreactor process. We've got the purification on protein A that's very standard for antibodies. You go through downstream polishing steps, and then you go through your UFDF steps. And what it turns out is that in the bioreactor, this is the biggest area where we can make gains in reducing the cost. And the way that's been found out, or what Fernando Garcia has modeled, who's in our group, is that most drugs, as I was talking about, they're in the range of $150 to $300 per gram, and that's just what they are. And as we said, Gates Foundation, that we work with very closely, they said you've got to get down to $20 a gram. We're like, God, how are we going to do that? Although we told them we could, and we're on the way. So what you look at is to look at the volumetric productivity in the bottom versus culture duration days, and the question is how to get there. And you can get down to about $50 a gram, and you look at how is that related? Well, that's related to how much material over on the right-hand side you're producing in this 500-liter bioreactor. So as you can coax your cells to produce more and more material, where you're getting up to 3 to 4 grams per liter per day, you can actually get down to the point of producing material that may be $20 to $30 a gram, and there's line of sight of how to get there. And then the last thing that you want to do is, well, that's all great. You need to produce low-cost manufacturing plants. And so we've actually done that, and this is actually the top one is our plant. This has a plant with a 500-liter bioreactor in the back. This is the purification. This is the exact same configuration that would go into a full manufacturing plant, and that we've actually produced one. Actually had one built in Hangzhou, China, and we've modeled that out, and you should be able to produce a metric ton of material per year out of these very small plants that you can actually build now for less than $100 million. And to give you an idea, to make a normal standard manufacturing plants these days are $500 million to $750 million. So this significantly lowers the cost and actually get into manufacturing and to maintain these. So that together is really what we've been trying to do and what Just has, and we're well on the way, hopefully, doing that. And you can see how all these parts are starting to integrate together. And I've named a few of the people here from Just, Randy Ketchum and Jeremy Shaver, and have really done a lot for the in silico modeling, and then the group that's done all the biophysics. But really, we have about 100 people now upstairs, and it's really everybody together has allowed this to happen. And then our external partners, the Gates Foundation, along with Michelle Nussensvig at Rockefeller University, and Dan Baruch. Thank you.
Thank you. We have time for a few questions.
So very interesting. Quick question on your attempt to improve the biophysics and stability. And we don't know everything why antibodies work. And could it be that there's an interference between increasing the stability and then affecting the biological activity, such as the FC part of things and so?
Mm-hmm. Yeah. So we pay a lot of attention whenever we did make modifications. We're always looking and testing the potency of the molecules, generally just for binding to them. So that's of prime importance, because we've said we can't knock that out. But we are starting to realize, and this was some of the data I was showing, that modifications that we're making up in the FAB region appear to either be translating down to the FC region in terms of the half-life, or possibly it's the stability in the bloodstream that's relating to the half-life. But we've actually done it now on 3 different molecules where we see these increases in the half-life, and so we've noticed that. We didn't expect it, but we're trying to understand that.